Okay, it looks like this. So this, oh, this is gonna crash. Stupid Adobe. Looks like this. You'll see that there's a specific example of the antibodies we're talking about today. And this goes through the step-by-step of comparing like the, yeah, see, that's what happens now. My Adobe is broken. On the left side, what you'll see is the generic sort of how monoclonal antibodies are produced. And then on the right, you have this specific type called Herceptin, and I'll show like a quick video. I might not show the whole video in lesson, but it's on iLearn. And then you're gonna like compare the general step with the specific step in the Herceptin production. Okay, so I'll just minimize that for now. And then you have down here the mark scheme. So the mark scheme here is for not only the question, the file I just showed you, but also for these two from yesterday, the vaccination IBQs, okay, and the data analysis IBQs. So all three questions, the mark scheme is here in this file. Okay. No problem. Is anyone alive out there? The least you could do is say, okay.
Okay. Okay.
Very disheartening to speak to nobody. Imagine if we were in class and I was teaching at the front of the class and you guys just ignored every single thing that I said.
We're not ignoring, we're just not responding.
Yes, but that's what it feels like to me.
We'll acknowledge you more often.
Yes, please. Like, I feel like, it feels like if you were just in class and just staring at me and I was like, everyone okay? And you just continued to stare, like not nodding, not saying yes. It's like, sweet, okay. Anyway.
Sweet.
Okay, let's get started. So last lesson for 11.1.
Wait, this is HL?
HL. We've been doing HL the last 3 lessons. Last 2 lessons.
Like for this one, because I remember you said like Monday, Tuesday. Oh, today's Tuesday. Okay. Oops.
Tomorrow. Tomorrow,
yes.
so. Okay, so Richard, you asked a question yesterday about white blood cells. Do you remember what you were asking?
Did I?
Yeah. You had something like, why can't you use white blood? Something about using white blood cells.
Why can't you just inject white blood cell into the thing instead of?
I tried to look it up. I have a feeling I couldn't find like a straight answer. It seems like it's not a thing. So there must be something I'm missing, but I think there must be some kind of like a rejection factor. There's a reason why you can't just take someone's white blood cells and then give them to someone else. Maybe a similar to like a blood typing, but more serious, and it just doesn't work. However, what we're learning today is a technique that we can almost use the same idea that Richard had, but not quite the same as using the entire white blood cell, but using their ability to make the antibody that we want, okay? So today's whole lesson is gonna be about monoclonal antibodies, okay? So this term is gonna come up. I would say like, your main goal is to be able to essentially answer like a 10-point story of how can you produce monoclonal antibodies. So if you keep that in the forefront of your mind, that's really the point. We're going to give some examples. We're going to go through, like I said, the treatment with the video, but really what you need to know is how is this done in a lab, okay? So first things first, what is a monoclonal antibody? Essentially, this is an antibody that specifically binds to only one antigen. And the reason we might want this is for either for testing, like for some kind of screening to see if, if we want to detect a certain antigen, or more kind of relevant or easier to understand, is for treatment. So imagine you have an illness caused by a certain pathogen. Wouldn't it be great if you could just produce, like mass produce, tons of antibody against that antigen. And then you could inject it as like a serum. So that would be like a passive, right? Passive form of immunity. But in order to make that serum, in order to make that medicine, you need to produce lots of antibodies somehow. And it has to happen outside of that person's body. So what we're referring to when we say this, when you say monoclonal, is we're making clones of one single antibody only, right? So a singular type of antibody that has a singular antigen that it matches to, but we're going to make lots and lots of it. Okay, so the two uses are for therapeutically, so meaning treatment, or diagnostically, meaning if you're scanning someone's like blood sample to see if they have a certain antigen. Okay, so here we go. This slide is going to give us the step-by-step process. So let's imagine we have an illness. Okay, we have a pathogen. Okay, that we know, and we want to create essentially let's call it a cure or like a treatment for it. We need lots of antibodies, right? So how do we get, oh, Janaki, I need to let Janaki in. Oopsie. And Amanda. Usually it gives me an alert when that's happening. Sorry, people. So we need to like trigger the white blood cells, the lymphocytes, into producing antibodies, right? But we don't want to be doing that like in the body. We don't want to inject the pathogen, like this harmful pathogen, into someone's body. We want to just create lots of antibodies and then put those into the person to create a treatment. We're not looking at long-term immunity here. We're looking at short-term. So how do we make a lot of copies of one antibody that we're interested in? First, we need to know the antigen that we're interested in, right? So this is the bad guy right now. We know that on the surface it has these proteins. Okay, fine. Since we're ethical to an extent, we're ethical to an extent. We're not going to use humans. We're going to take a mouse, okay? We take this mouse and we're going to inject the antigen into the mouse. So what is that going to do in the mouse?
Kill the mouse.
Hopefully not. What's another option?
Active immunity.
So it will trigger an immune response, right? If you inject any animal with an antigen, we're basically vaccinating, right? We're vaccinating this mouse. It could be that we don't give him the live pathogen. Maybe we just give him like a sort of like a vaccine. So what the mouse does, it goes through the specific immune response. And the specific immune response, what's the result? Same as normal. Plasma cells that produce tons of antibodies, memory cells, blah, 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 whatever. What we want are those plasma cells, right? Those plasma cells, the ones that produce the antibodies. Those are the ones that are really important to us. So what we'll do is then we need to screen the sample to find the plasma cells that we're interested in, okay? So we're gonna take the plasma cells out of the spleen of the animal. The spleen, the spleen, because that's where we have a lot of white blood cell production, okay? So you'll find a high concentration of white blood cells there. So it's a high likelihood that you find the one that you're looking for. And then you're going to, and this is how we can make many, many copies of this cell that we're interested in, okay? There's a part in here that's not mentioned. I should put it in here, okay? Which is screening, okay? So screening of the sample to find the one that you want, right? The plasma cell that you want. So you only choose the one that that matches the antigen that you're interested in. And this is where the really cool part comes in. In order to make many copies of this plasma cell, we're going to take this cell and we're going to fuse it, okay, with literally a cancer cell. Okay?
But why?
Oh, wow. Okay, interesting.
So you take this cell and you fuse it, meaning you just shove them together. And we know the properties of cell membranes, right? They can reform and like they can join together. And so it's all made of the same stuff. And now you've created, and this is a really important term here, you've created a cell called a hybridoma, okay? Hybridoma cell. The oma part is referring to the cancer, right? We know like melanoma is a type of cancer, right? And hybrid meaning a mix of two things. So this hybridoma cell is a combo B lymphocyte, like a plasma cell that produces antibodies, but also a tumor cell. Now, what's the property of the tumor cell that we are interested in?
Unlimited, how do you call that? Mitosis?
Right, not unlimited, but
multiplying itself, like cloning. No.
yes, but I'm looking for one more like key.
Binary food. No.
Yes. Like a description. It does binary fission. It does mitosis. It does it very
Quickly.
fast, like frequently, right? Cancer cells, the problem, why it's a problem, is because they divide so quickly and out of control. Usually that's a bad thing. Like, if it's going to make a tumor, then you like that this is a problem for a person. But if you now fuse it with this plasma cell that you want many, many copies of, now that those aspects of the plasma cell, like antibody production, we're going to make tons of copies of this new hybridoma, this blended white blood cell cancer cell. What is the result that we get? The result is we then screen, I should say, there's another screening process. So we can move this screening to here, okay, to make sure we're looking at the correct one, the correct type of cell, because we don't want some random plasma cell that we're not interested in, to find the one that's producing the antibodies that we want, okay? Which one is producing the useful antibodies? Once we know that, we pick out only that cell, the one that's making the antibodies we are interested in. And then we just put it in a perfect condition where it can divide until infinity, right? It will just keep dividing, keep dividing, keep dividing. And we can then, lyse those cells, burst them open, and take screen out, or like kind of it's like a filtering process, sort of filter out all the antibodies. And now we have this huge stash of these these antibodies. Come on. These antibodies that we wanted, which we can then inject into a person who might be infected by this pathogen. And those antibodies will go and do any one of those 4 things that would normally be done by your immune system, but much more slowly.
How do they fuse the plasma cell with the tumor cell?
Honestly, like that. It's really.
But isn't it inside the mouse?
But
you take it out of the mouse. You take harvested means to remove. So you actually take out a huge sample of white blood cells from the spleen of the mouse. Then you add them to a dish with the tumor cells, force them to fuse together. Then you look for the ones that you're interested in.
Does the mouse die? Like after the plasma cells are removed from the spleen.
I think
I think.
it depends on how the
What
antibody.
is a spleen anyways?
Huh?
What is a spleen?
Spleen is an organ that's part of the lymphatic system.
Oh, great. So you're removing, okay. So you're basically killing the mouse really, really slowly.
You don't have to necessarily take the whole spleen. I guess you probably do. To answer your question, Yushan, probably the mouse will die. Unless you only inject it with like harmless antigen, something similar to a vaccine. You could do the same thing by injecting it with like attenuated weekend, right? We learned that last time.
So the mouse either produces plasma cells or it just dies from the antigen.
Yeah, if you inject it with the real pathogen, the whole pathogen, then I guess it might die. If it's a deadly pathogen, it might die. But hopefully, they're just using the antigen itself that's weakened or damaged, so that it doesn't have its full strength. The point is, and you bring up a good point, right? Ethics of animal research. Yeah.
Mr. Nilan,
Mm-hmm.
then the hybridoma cells is gonna have two nucleus, one from the plasma cell, one from the tumor cell. Which one is gonna use?
Both. Probably both.
But the DNA is gonna, it divides through mitosis, so the DNA is gonna divide itself also.
Oh, wait a second. Hold on. No, it has to be both because you can't remove any of the information. That's an interesting question. And I also feel like I've already looked at the answer to that and I've forgotten it now. Maybe see if you can find, does a hybridoma cell have two nuclei?
Mr. Nilan,
Yeah.
it says hyperdoma cells are screened to determine which one are the useful ones. So does that mean not all is useful?
By useful, they mean like the one that fits the antigen that you're interested in, right?
But isn't it like if the plasma cell has the antibody, wouldn't it be all the ones you're looking for?
But when you remove the cells from the mouse spleen, you're going to get all kinds of different lymphocytes, right?
Oh.
So at that time, you're going to have like a whole selection of lymphocytes because the spleen has like tons of white blood cells. So it will have the one you're looking for, but also many other types. So then you have to like get rid of the ones that you don't need and focus on only the one that has the antibody for the antigen that you're looking for.
Okay.
Okay.
Rio is up.
Yeah, he's yeah. So this is a nice little sort of a cute summary of how this works. So our little mousy friend here. Okay, stimulate his antibody production. So make him start producing or create clones of the plasma cell you're interested in. Find the cell you're looking for. That could happen at this stage, like I said, or later on. Fuse it with a tumor cell. Allow them to divide rapidly. Screen them for the ones that are producing the antibodies you're looking for. And then clone them many times. And then isolate the antibodies you need. And now you can just inject this directly into someone who is ill with that pathogen that has that antigen that you started with over here. Remember though, this is a this is a form of passive immunity, right? Injecting only antibodies does not give you immunity, right? No immunity. So this is short term.
But then the plasma cells that like what is formed in the mouse's body, that's active immunity.
Or could be, yeah, could be. If they produce memory cells, which they should, then as long as you're not actually killing the mouse and using an active pathogen, if you're using a weakened one, then ideally the mouse would get vaccinated, right? It would gain immunity. Yeah.
That's a really big needle.
So the idea here is like these things would be used for like a really like a treatment that you need like quickly, right? So the example earlier is for rabies. Like if you get bit by a rabid dog or a rabid animal of any kind, you need to act quite quickly because the what do you call it? The rabies virus moves fast. Like every day it progresses like a certain distance, and if it gets like to your brain, then you're in big trouble. So you don't have time to just inject like a vaccine and then wait for your body to figure it out and go through the process. So that's where you would need something like this, where you'd need concentrated, just antibodies, get them in there, fight the fight, and then, you know, deal with it before you actually get sick from the situation. Okay, from the infection, I should say. All right, we're gonna go through wait a minute, hold on. I gotta see something here. Oh, it's not there. Okay, I'm gonna show you what this video is. Okay, this video is what's gonna help you with these IBQs. And I'm not gonna show the video right now. It is a very, very interesting video. So what it's about is a cancer treatment called Herceptin. Okay, so it's a breast cancer treatment, and the treatment uses monoclonal antibodies. So what it's doing is it's targeting the specific cancer-causing cells with an antibody. So they've been able to find the antigen shape, and then they've produced, like mass-produced, this HER2 antibody. You can see, I'll just kind of scan forward. You know what? It's only 3 minutes. Maybe I should just watch it. No, it's okay. So they are able to target the tumor cells, right? So there's the HER2 receptor, and then they've been able to isolate this Herceptin antibody, right? And then it goes and binds, and then it targets those cells for destruction. It's really like cutting edge. Like right now, these are the types of treatments that are being used. I forget the statistic exactly, but it's gotten to a point where like breast cancer, which is like one of the highest, has one of the highest prevalences for cancer in women. It's almost, I'm not downplaying, but it's almost like not even that bad if you catch it early enough because of how many different treatment options there are, including this one. So there's a lot of people that are, you know, being diagnosed with breast cancer, and they're coming out of it before it gets too severe, because of a lot of developments in the treatments that are available, and one of them being this monoclonal antibody treatment called Herceptin. So definitely check out this vid, and it'll help you answer these questions. But without going into that, we're going to look at a different example that I think is pretty cool of how monoclonal antibodies are used in a pregnancy test. Okay, So does anyone know what this movie is?
It has
No.
Howard in it
Howard.
from The Office.
Dwight?
Oh, Dwight, yeah.
Dwight Howard? Yes, it does have Dwight from The Office. No? Okay, this is a movie called, and if you want a really fun movie to watch, it's called Juno. Juno. Anyone?
Nope.
It's really good. Basically, it's like this teenager gets pregnant, and it's like not serious. She's like very jokes about it, and like very quirky, very funny. Sort of how she deals with the rest of her situation and being pregnant and deciding what to do with it. But the point being, there's a scene where she uses a pregnancy test. And I'm going to just go through quickly because this is one of the applications of how monoclonal antibodies are used in a pregnancy test kit. So, nine months ago, oh no, I hope I didn't actually click on that. Oh, bloody hell. What's going on? Oh, my PPT is frozen. I'm out. Hello. Oh boy. Okay, give me a second to troubleshoot here. This is fun. Okay, we're just going to jump to this. So, pregnancy test works like this. So you might have seen this in the movies, right? Does anyone know how this works?
Mr. Nina is actually secretly testing us right now. If anybody says yes, he's gonna be like 911.
I'll assume your knowledge is from media. How does a pregnancy test work? What needs to be done?
Good line.
I think the female user of this device urinates onto the device.
Right, precisely. Okay, so the point is you pee, right, or you urinate on the stick. It is the female. She's the one who needs to urinate on the stick, to be clear. Yeah, 9 months ago, Miss Katie showed me one of these things, and I was like, oh shit, here we go. Anyway, how it works is the following. If a woman becomes pregnant, meaning that her egg has become fertilized, okay? Right, we know this basic understanding, sperm, egg, bam, okay? And then it starts to divide, and we have an embryo. That embryo, that little ball of cells that's starting to divide, it produces a hormone, okay, called HCG, human chorionic gonadotropin. There's my highlighter.
ACG.
H.
Animal chromo. Something.
Okay. Human chorionic gonadotropin. Okay, so this hormone
Do we need to put the name?
Sorry?
We need to know the
No. Just HCG is fine. HCG is fine. Okay. So HCG is being emitted, okay, by the little ball of cells that's dividing. So meaning it's in the mother's bloodstream. Okay. Even if she doesn't know, she may not have any idea, but it's being produced by the embryo. Okay. So that's step one. That's going on. If this is a pregnant lady, she has this in her bloodstream. Now to the pregnancy test. Okay, the pregnancy test is this like plastic stick with a strip here, sort of like filter paper, and it has essentially two little windows. Okay, on the test, and you can see here this is the legend, right, or the key, meaning if you see a line. If you see a line in this window and this window, then you are pregnant. If you see a line only in this window, then you're not pregnant. So the question is, how do those lines appear, right? How do we get those lines? And what we're going to see is that this has entirely to do with monoclonal antibodies, okay? So we're gonna see the link. What's the link between HCG, the antibodies, and this blue coloration, okay? So here, you've got 3 lines on the pregnancy test to start with, right? Before there's urine. So you have first a line here that is a combination of antibodies for HCG, meaning it's an antibody that matches with this hormone. Okay, so it's been mass produced, right? Just like before with the monoclonal antibody, inject the HCG, then this antibody matches, blah, 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 make lots and lots and lots of this. But it's connected to, it's been like attached to this blue dye. That's what this little box is showing. Okay, so it's an HCG antibody attached to blue dye. Further down the line, you have this strip, and at first, this would be white. It doesn't show up, there's no color to it. This is just antibodies for HCG. So meaning, what can attach to this? HCG, the same as this one. Okay, this and this are the same. Now, the last line is antibodies for blue dye. Okay, so there's another blank line here that doesn't have any color. And it has fixed onto the paper, onto the strip, these antibodies for blue dye. So the whole idea of antibodies is to attach to their proper antigen, right? So we have antibodies for the dye, antibodies for the hormone attached to blue dye. So can we, without clicking ahead, tell me, how does this work? The woman pees here on the end of the pregnancy test. And since this is like filter paper, right? The urine starts to use capillary action to flow this way. Tell me what happens if the woman is pregnant.
The antibodies are gonna stick onto the hormone and move along with the urine, with the dye, I guess.
Okay, so it's gonna grab this because if she's pregnant, she has HCG, right? And then it's going to keep moving. So these guys keep moving. Then what? So let's use this. Let's use a different color and say HCG is like a green circle, right? This is matching like a circle. Okay, so then that's going to move. Now what?
Maybe it's gonna keep moving to the right.
Okay.
And then the antibodies for HCG is gonna continue stick onto the antigens, making it heavier, which is not gonna move. I'm not sure.
Close, close. Kind of. What can we do with this shape here? So it's basically a square with the antibody and the antigen or the hormone is here.
The hormone gets trapped in the first layer.
The hormone can attach the other side of it here. So what does it do? They get stuck there. And since there's a blue dye, I should have drawn that in blue. Since there's a blue dye here, you get this line being blue. Okay, so that explains this one. What happens down here?
I have no idea.
How come if the woman's pregnant, how come you'll see both lines?
Because some are stopped at the first layer, some are stopped at the second layer.
Yeah, so not everything gets stopped here. Some of it will continue on and it will get attached to this one. And I'm drawing this very crudely. And it'll be there like this. So then you'll get some of it that keeps going to here. And you'll get a line showing there because the blue dye made it here. You'll get some of the blue dye that got stopped here. So you end up with two lines.
But the second layer here says like here antibodies for HCG attached to blue dye. What if the urine doesn't have HCG?
Okay.
Wouldn't that not attach?
Right. So let me clear this stuff and let Joseph into class. I'm not sure why he's trying to join class, but okay. Yes, so that was the example if she is pregnant, right? So let's look. Pregnant. Urine sample with HCG hormone. Okay, step by step. 1. HCG molecules in the urine attached to the anti-HCG antibodies. And as you can see, like some stuff leaks through, right? Some of it will attach to the first strip here, whereas some will continue on and attach to the second line, giving you the two lines here and here. Now, as Michael asked, what happens if you're not pregnant? So here's the urine with no hormone in it. So the urine flows by. When it hits this line, the blue dye will still move, right? But this time it's just not attached to any hormone. So the blue dye is not like fixed on the strip. Only these ones are fixed on the strip, these two. So the blue dye moves. It doesn't stop here because there's nothing for these anti-HCG antibodies to attach to. So they move all the way past to the antibodies for the blue dye and they stop there and you end up with only one line. And depending on the situation that you're in, you would see one line and say, yay, or no. Or you might see two lines and say, oh, yay. Or you might think, oh, no. You know, currently, oh, no seems like a better option. Yeah.
Is there by chance the stick is wrong?
Is there a?
Like by chance, like for example, you're not pregnant, but it shows two.
Hopefully not. So that's a good point. So there's really, these pregnancy tests are very, they're not like super sensitive, right? They're more like a starting point, and then you obviously go to see a doctor, and then they would confirm it for you, either by ultrasound or by a more sensitive blood test. But yeah, there is a possibility. If I'm not sure what other sources there are for HCG, but it's pretty closely linked to embryonic development. So that's why they can use that hormone as the one. False positives are always possible though. And these pregnancy tests cost like not thing. So it's not like they're made to be very, very, very discerning.
Okay.
Yeah, good question. So yes.
What if a male urinates on the stick and it shows 2 lines?
Hmm? Evolution?
I don't know. I think there's something else going on. Producing HCG from something else. I mean, at the end of the day, like, we're also making a leap, right? This is just a test. If you didn't call it a pregnancy test, you could call it an HCG test, right? You're just testing your urine for this hormone. So you then just make the link between the fact that pregnancy releases HCG, therefore, blah, blah, blah.
But
So, yeah.
why is the second window important to be shown? Anyways, it's gonna have blue colors always.
Because I mean, the blue dye is it has to stop somewhere, right? I mean, I guess you could just run it to the end, but then this tells you that it worked essentially, right? Like, it's like you know that the dye still moved. So if there was a chance that it was positive, you know that it passed that line, the window in front of you, and you know that it worked. It got to the second part, so you know for sure there's nothing there. So it's like it tells you that the test worked.
Okay. But why does the molecule stop at the first line? Like, aren't supposed to keep moving along with the urine, even the antibodies are attached to it?
No, because
What?
because these antibodies and these antibodies are like fixed. They're attached to that point. They don't move. Only the first section where the dye is there, that part is like not attached to the paper, or it's like let's say it's like mobile, or yeah. Whereas these ones are like immobile.
Mm-hmm. Okay.
Okay, so aside from treatment or whatever this pregnancy test, there's another reason why we could use this. It could be used for screening, right? So if you're testing to see if someone has an illness, so for example, like the nucleic acid tests that we would use if you're testing to see if someone is positive for COVID-19, might use this kind of a test. So you have the antibody, you screen this person's sample to see if they attach to the antigen, ideally with some kind of like fluorescence or some other like indicator, like something that you can see under the microscope or under fluorescent light. So you can attach like to the antibody, you can attach little markers that help you to see where it is when you look at it in different conditions. And then the very last part of this topic is about epidemiological analysis. Now, in normal years, we would spend time on this, explaining what this all means, but I'm pretty sure that over the last 3 or 4 months, we have a pretty solid, we've been sort of bombarded with the idea of epidemiology, with the idea of, studies and collecting data and how we track diseases and so on. You see it in the news, you hear it in the news, you read it, on the internet. In this example, of course, it's talking about measles, which is, like we said last time, an illness that we get vaccinated for, but it depends on like what countries you're in and how reliable the vaccinations are, or how many people actually do get vaccinated. So this area of study is really, really important, and we know this now, that this is a critical component of not just a country's research in healthcare responsibility, but also a worldwide responsibility. You can see here, so this is really, you know, very relevant to us right now, is that most member states of the World Health Organization submit monthly reports on cases of measles. And this is just one disease, but they would report on any of these types of like very contagious diseases, right? So that the WHO knows, for example, like, hey, this country is showing like the numbers are sort of like going up, whereas they haven't been doing that. Like it's normally the stable trend. And then maybe the World Health Organization is looking for like trends or clusters or something. And obviously like, there's many things that we could say, okay, about monitoring and, you know, responsibilities for monitoring and accuracy in reporting of cases of illnesses. I think you know what I'm referring to. And if not, don't worry about it. We can talk about it some other time. But this idea is, this study of epidemiology is so critical for global health, and we're seeing it right now. The response, thankfully, has been quite strong. So the world is getting on board. They're sharing information, and they're able to now like share all this data and sort of fix it. But it also should be able to prevent any serious cases from taking place by, you know, keeping track of your own country's information and then submitting it to a higher body who can notice trends and look for any issues that might occur. All right, so this idea of like national disease surveillance, which China has, right? They have a, yeah. I'm gonna stop there. So then you might see, you know, this kind of data, and actually this is a really, the WHO has lots of data of many different diseases, and it's sort of incidences across the world, right, in different countries and regions, as well as across time. This might be something you might use if you wanted to do like a secondary research II next year, when it comes down to it, when you have to pick your own individual investigation. Yeah, so keep that in mind. There's lots of databases with lots and lots of data available to use. And using that graph, you might want to try this as a data analysis question. So like I said, this topic has lots of potential to have a question show up because there's so much data available, right? So you might see, the answers are there, I don't want to show you, but you might see like graphs like this, and then they ask you to analyze the trends, right? Annual pattern, what region is the most impacted, et cetera, et cetera. Okay, so I recommend you try this, or at least this one, or if not the other one that's on iLearn for you. And that's it.
Yay.
It's the end of 11.1. So this one has a lot of work attached to it, guys, or a lot of IBQs attached to it. So you've got IBQs here, data analysis here, more IBQs here. This one links to this video, and then data analysis from the PPT. It's a big topic. Definitely one where the data analysis could be a key factor. So, any questions?
Nope.
Everyone's fine.
Hi.
Hi. Let's stop.